of energy balance across the overdrive and healthy subjects.
Good morning, members of the RECNET. My name is Xu Jiapan. I'm a Chinese researcher from Shanghai Regional Hospital, affiliated to Shanghai Jiaotong University School of Medicine. Due to COVID-related travel restrictions, we won't be able to make it in person this time, but still, I would like to share our study on the effects of time-restricted feeding on energy balance with everyone here. So now, first of all, let me take this precious opportunity to introduce our lab to the world of science. So Shanghai Regional Hospital has established the world's largest digital metabolism research center with in total 10 whole room indirect calorimeters. The center has constructed a topological network to demonstrate a unique continuous monitoring system of human-environment interrelationships. Our lab is also a digital upgrade of the traditional metabolic cabin, as it consists of 4 independent monitoring systems, including the environment, metabolism, behavior, and physiology, along with the traditional dynamic metabolic data like the RQ and EE. All environmental, physiological, and behavioral parameters of the subjects are also being collected seamlessly, generating a high-dimensional time series data in real time for the analysis of research on human energy balance. Here we emphasize the word environment because we believe that environmental exosomics play such an important role in human metabolism, and our environment control includes independently controllable temperature and humidity with the advanced illumination system simulating a range of different color temperatures. And in addition, our lab also has the world's first hypoxic metabolic chamber, which not only has all the functions of a standard chamber, but can accurately simulate different altitudes by reducing the partial pressure of oxygen using electronic scrubbers. And this will allow determination of energetics of the human body at different altitudes. And this is also the designated unit for high altitude metabolic research by the China National Health Commission. So back to our study, we all know that energy metabolism is one of the most basic features of life and is always in dynamic development. It changes with different genes, races, body composition, with aging, growth, reproduction, diseases, exercise, and of course with daily habits and lifestyle. During the past few decades, the prevalence of obesity and related chronic diseases has increased dramatically, and the current first line of therapy for overweight and obesity is aggressive lifestyle modifications, including reducing calorie intake, improving diet quality, and increasing physical activity. However, long-term adherence and maintenance to such strategies are challenging. Time-restricted feeding, TRF, is a specific intermediate fasting protocol that has gained attention as a more acceptable and feasible regimen. And this study is designed to systematically explain how TRF creates an active energy balance without reducing calorie intakes. And this is our study design. In total, 159 subjects are being assessed, and 12 has completed this randomized crossover study. And this is the study design of the research. The daily schedule consisted of 2 patterns, the control and TRF, and each dietary intervention was confined to the metabolic chamber for 3 days, during which calories of each meal and physical activities were rigorously controlled. As a crossover study, there is also a 1-week washout period between the 2 stages. For the control study, the first meal was taken at 8 a.m. and the second one at 1:30 p.m. with the third one at 7 p.m. For the TRF, all 3 meals were finished within a 5.5-hour eating window. In particular, all foods and feces were labeled in brilliant green dye and were collected, mixed well, and lyophilized for energy and contents evaluation. The 24-hour urine were also collected on the intervention day. The protein, fat, carbohydrate concentration were quantified for both the food and the feces. In terms of 24-hour energy expenditure, we found actually no significant differences in mean energy expenditure due to TRF, and the only difference arises as a result of postprandial thermic effects of food. However, surprisingly, in fecal analysis, we found that the total fecal wet weight and fecal energy loss has increased significantly in the TRF group. Specifically, the fecal energy loss has increased by 22.7% in the TRF group, which includes an increase in fecal carbohydrate loss, fecal fat loss, and fecal protein loss. It is known that around 2 to 10% of food consumed ends up being excreted in feces, and even if nutrient enters the circulation, there is still a 1 to 2% of total energy intake finally being filtered by the kidneys. The precise measurement of the third and often neglected factor, the energy expression, finally helped reveal that TRF has resulted in a negative energy balance of negative 46 kilocalorie, which is equivalent to 2.6% of total energy intake as a result of increasing fecal and urine energy loss. In terms of non-proton RQ, we found that TRF increased NPRQ nearly continuously across a 7-hour period from 12 to 8 p.m. and that the substrate balance significantly shifted towards carbohydrate in TRF group with better metabolic flexibility. In terms of blood glucose level, it is seen that the average glucose over a 24-hour TRF intervention was reduced by 5%, which was largely contributed by the extensive attenuation of postprandial glycemic profile in TRF group. The glycemic variability was significantly blunted during TRF intervention as reflected by the significantly lower coefficient variation and mean amplitude of glycemic excursions. Since glycemic variability has been demonstrated to be closely correlated to oxidative stress and beta cell functions in previous studies, such an extensive attenuation of both glucose fluctuation may help to explain the long-term benefits of TRF for glycemic control. As regard to the physiological profile during TRF, we observed an obvious cardiovascular and respiratory-related thermogenic process, and this is there is increasing evidence that increased heart rate is associated with hypertension and mortality, while decreased heart rate could help improve this situation, and this is another advantage that we found out of TRF. So at the end of this presentation, I would like to thank my mentor, Professor Ning Wang and Wei-Chun Wang, as well as my research team at Shanghai Regional Hospital. I'm so proud of them. And also I would like to say thank you for Dr. Russell Rising, who is the honorary consultant of our lab. And thank you very much for listening. Do I have any questions?
Thank you very much for this clear presentation. I think there are questions, please.
Dr. Yamauchi, first of all, I'd like to congratulate nice work. And it is an eye-opener that you focus on urine and fecal energy loss. My question is about RQ urgency. In your experiment, RQ continue to increase urgency instead of fasting. And what is your comment about it? Because I'm asking you that I am using the same system in Japan.
Yes, actually it's quite an interesting finding because in both of our groups, like the control and TRF, the RQ keeps increasing during sleep. And this is probably because of our research settings. I don't know if you explain that, but that's what we found. Do you have any similar observations in your lab?
Yes, similar, and it depends on gender. But
we didn't find any gender specificity in our study here.
Thank you very much.
Thank you.
Question?
Eric Robinson, we did a very similar study in the chamber a few years ago, and we found a decrease in the 24-hour respiratory quotient, which was significant and quite not negligible. Do you think it's just because the shortness, it's really the first day or the second day I missed the design of your TRF that you do the measurements? We did that after 6 days or 7 days, if I recall correctly, it's published. But do you think it would make an impact on a longer term on RQ and therefore fat oxidation?
Yes, of course, that's a very good point. I think the interesting, I think it's very important to have different observations from a similar study, especially when we change one specific factor of the intervention. For our study, it's the very first day that we observed for the TRF study. However, we do have some very primary data of longer terms, and we did find similar things like the RQ. It starts going on a different direction when the TRF intervention gets longer. It's probably because the body gets a new balance after a long period of intervention. And that's very interesting.
Thank you very much. If there are no more questions, sorry, please.
That's a very nice study. Thank you very much. So I'm wondering the difference with regards to fecal loss of energies. It seems quite striking. I was just wondering whether you could comment on the possible role of the change in chylomicron secretion. It's well known that with repeated meals, you secrete more and more of the fat that was absorbed from previous meals. So I'm just wondering whether you have thought about these possibilities with regards to the change in intestinal handling of energy metabolism.
Yeah, that's very interesting, and that's actually another study carried out at our lab. So probably we can answer this question in the next Reichman 3 years later.
Can't wait to see that. Thank you very much.
Okay.